Hi everyone. Tonight we're going to talk about the molecular basis of inheritance, or the structure and function of DNA. Our objectives are to demonstrate an understanding of how cells use DNA. I discuss transcription and translation in a separate video this week, but today we're going to be focusing more on the structure and function of DNA itself. So what is the structure of DNA? Well, according to Chargaff's rules, which if you remember, Erwin Chargaff did this really cool experiment where he extracted DNA from many different widely varied species, and he kind of put it in a blender and was able to separate out the adenine, the thymine, guanine, and cytosine, and he divided them by percents. And what he found was that the percents of adenine always equaled or just about equaled the percent of DNA that was made up by thymine. And the percents of guanine usually just about matched up perfectly with the percent composition that was made up of cytosine. So basically what this boiled down to was every time there was an adenine in the DNA, there had to be a thymine to go with it. And every time there was a cytosine, there had to be a guanine to go with that. And this is how we figured out the base pairing rules, that A and T always bind together and G and C only bind with each other as well. A trick to help you remember this is apples in trees, car in garage. I would write that one down. It's easy to get them mixed up otherwise. All right, so another famous scientist, Rosalind Franklin, in the 1950s, took this famous picture. It's an X-ray diffraction image of DNA. And really, the best way to look at this is to think of it as if you were lying on your back on the floor, looking up through the double helix of DNA. Or kind of like if you're at the top of a spiral staircase looking down, what you might see. She worked with some other important scientists together, like Maurice Wilkins, to produce this image of DNA. It's called X-ray crystallography. Is the type of imagery she used to get this picture. And she provided measurements on the chemistry of DNA. What's its chemical makeup? James Watson and Francis Crick used this image and the data gathered by Franklin and Wilkins. And they are the ones that riddled out that DNA has to be arranged in this double helix shape. All right, so I've said the word double helix. It's like a spiral staircase. The outside of the spiral staircase is made up of this sugar and phosphate backbone. So in these models, the sugars are blue, the phosphates are yellow, right? So we see there's blue and yellow along the outside of the strands. And then the rungs of the ladder, or the steps on the spiral staircase, are made up of the nitrogenous bases, the thymine, adenine, cytosine, and guanines. So, nitrogenous bases, we have two purines, adenine and guanine. These are both made up of double ring structures. And thymine and cytosine, these are the pyrimidines. They each are made of a single ring structure. And here we have our pairing rule. So adenine and thymine only bind with each other. They make two hydrogen bonds. Guanine and cytosine only bind with each other, and they share 3 hydrogen bonds. They fit together like puzzle pieces. You can't have a thymine and a guanine bind together because they don't match up. Thymine needs 2 hydrogen bonds, but guanine needs 3. So they only bond in this very predictable fashion. All right, hydrogen bonds are the bonds between those base pairs. They hold the molecule together a lot like the way a zipper works. DNA strands are anti-parallel. That means one strand runs from a 5' to a 3' end, And the other strand runs in the opposite direction, upside down. So we got 5' to 3', up and down. They're numbered by where the phosphate attaches, which carbon it attaches to. So 5' to 3', and then the other one goes 3' to 5'. But see how it's upside down? And the way I check that is I look to see where these oxygens are. The oxygens are on top of the deoxyribose sugars on the 5' to 3' side, but on the 3' to 5', they're on the bottom. And we call this anti-parallel. How is DNA packaged? So we know DNA makes up a double helix. That double helix can be wrapped around histones so that it occupies less space. And then those histones wrap and scaffold more to structure themselves as chromosomes. So we've got one chromosome and then its replicate, in this instance over here on the left. A pair of replicated chromosomes is called, replicated chromosomes. Each single individual side is a chromatid. And we'll talk about those more when we talk about cell division and mitosis. But what's important to know is that DNA is a double helix, but when it wraps up around histones, it can make chromosomes. And this is just a more efficient way to package it, right? Think of like ramen noodles. When you buy a package of ramen and it's all wound up tight and neat and orderly inside the package in that little rectangle, it occupies way less space than it does after you cook it and you unwind all of those little noodles, right? Okay, so DNA is a double helix, but it can be wrapped up and packaged more efficiently as a chromosome. All right, so now let's compare prokaryotic and eukaryotic DNA. Prokaryotic DNA is double-stranded, as is eukaryotic DNA. Prokaryotic DNA is circular, though. It makes a circle. Eukaryotic DNA is linear. We have these linear chunks of DNA called chromosomes, right? There's one chromosome in a prokaryotic cell, but in eukaryotic cell, we usually have multiple chromosomes. Prokaryotic DNA is stored in the cytoplasm. Eukaryotic DNA is stored in a nucleus. We call supercoiled DNA in a prokaryotic cell the nucleoid. That's where it's just kind of clumped up together. In eukaryotic DNA, the DNA is wrapped around histone proteins to produce chromatin or chromosomes. There are no histones in prokaryotic DNA. All right, so we know that every cell has DNA, right? But when cells divide, how do we get more DNA for each cell? How does DNA replicate or copy itself? Well, this is super cool. So making DNA from pre-existing DNA. When biologists first tried to tackle this question, there were 3 different competing ideas. Some people thought that DNA replication was conservative, where you end up with an original copy and a new one, right? Some people thought, well, maybe it's semi-conservative. Maybe each of the new strands of DNA gets one strand from the original. And then some other scientists thought maybe it's dispersive. Maybe there's bits from the original that ends up in all the strands. Well, Mieselson and Stahl did this really cool experiment where they cultured bacteria in a medium with a heavy isotope of nitrogen in it. And they transferred the bacteria to a medium with a lighter isotope. So they grew bacteria, there was cell division, DNA was replicated with this heavy isotope. Then they transferred it to another place where there was a lighter isotope and let them grow. The DNA sample, so they sampled DNA from this, and they centrifuged it after the first replication. So they let the cells divide one time, and there's a single band of DNA. It means the DNA basically all had the same weight. The DNA sample is centrifuged again after a second round of cell replication, or after DNA was copied. And they found a second, less dense band. This is important because what it shows us is that it's not a conservative model, because we don't end up with a really big light band and a really small dark band, right? They were equivalent, which is what we see here with semi-conservative. If it were dispersive, we would expect to see something where, all the DNA would be about the same weight. So this is how we determine that it's a semi-conservative model of replication. After the first cell replication, one of the daughter strands of DNA has an original template and then a newly made complementary strand. And then the opposite strands are complementary and template in the other one. So we get half original and half new DNA. And then after those cells divide, we have some that are like this lighter strand here will go on to replicate and be made with, it'll be one of these that's 2 light copies. And then we'll have another that's half and half. So that's how we ended up with the 2 different bands in the test tube. All right, so replication is semi-conservative. We have a parent molecule, there's a separation of strands, and there's daughter DNA molecules, each consisting of 1 parental strand and 1 new strand. All right, there's a neat little video on here I want to make sure you watch. I will put a link to it in our D2L page as well, but do make sure you take the time. The animation is super short. I think it's less than 2 minutes, but it's a really great way to see the whole process in action. All right, so we have our parental DNA here, right? We see that it's being unzipped. The enzyme responsible for unzipping that DNA is called helicase. Next, we have DNA polymerase 3, which attaches, and then we have DNA replication that occurs. So there's some important enzymes in here: polymerase, helicase, and ligase. It must be on a different slide. But anyways, we'll walk you through it. So the major steps of replication. First, DNA helicase unwinds the DNA at the origins of replication. It's kind of like the start spot for copying DNA. Initiation proteins separate the 2 strands from each other, and this forms a replication bubble. So a bubble where the DNA is unwound. Topoisomerase relieves overwinding strain ahead of the replication fork, or ahead of the split, the bubble, by breaking, swiveling, and rejoining DNA strands. Primase puts down an RNA primer to start replication. DNA polymerase 3 adds complementary bases to the leading strand so that new DNA is made 5' to 3'. A lagging strand grows in the opposite direction by the addition of small bits of DNA that have to be joined together, and these are called Okazaki fragments. DNA polymerase 1 replaces the RNA primers with DNA. And the DNA ligase is the enzyme that seals the Okazaki fragments together. All right, so let's zoom in on each of these steps. Helicase unwinds DNA at the origins of replication and creates the replication forks. You can see those with the arrows down here in the micrographs. So in a bacterial cell like E. coli, we have 2 replication forks because their chromosome is a circle, right? And then DNA replication can move forward in both directions. And eventually we end up with 2 separate chromosomes, produce 2 daughter DNA molecules. The origin of replication in a eukaryotic cell is a little different because it's linear. So we have an origin of replication here, and we might actually have multiple origins of replication along the length of a chromosome. And that allows for DNA replication to happen in a much more efficient manner. So we have our replication bubbles going here. We have template DNA, replications proceeding in both directions. So the parental strand of DNA is the dark blue, and the new daughter strands are being formed in the lighter color here. Excuse me. All right. In the end, after these work and they join together, we'll end up with 2 complete daughter DNA molecules, each with 1 original parent strand and 1 new daughter strand. And the same is true in the prokaryotic side, right? Parental, daughter, parental, daughter. All right, next up, primase adds an RNA primer. Here's the pink primase, and these little red nucleotides here are the RNA primer. DNA polymerase 3 adds nucleotides in a 5' to 3' direction along the leading strand. So we have our template strand, and we're adding nucleotides 5' to 3'. You can see where each new one is coming on. A binds with T, C binds with G, A and T, and so on. We're building up the phosphate sugar backbone so we can add more nucleotide rungs to the ladder. Now, I've mentioned leading strand and lagging strand. Let's talk a little bit more about that. So here's our origin of replication. There's the little orange primer. From here, DNA can be created or produced a complementary strand in one continuous swoop, one continuous strand. And that's called the leading strand. So it goes 5' to 3'. We also have a lagging strand. And a lagging strand, since DNA can only be made 5' to 3', on the other side of the origin of replication, the DNA loops and it allows us to make short chunks in the 5' to 3' direction. These little chunks of replicated DNA, these are the Okazaki fragments. They'll have to be combined together to make one continuous strand and match up with the lagging or with the leading strand. So replication on the leading strand is able to happen in one continuous motion as helicase unwinds. But on the lagging strand, we have to go piece by piece and then fit all the pieces together to build a long complementary strand of DNA. So Okazaki fragments are the short segments of DNA that grow 5' to 3' and are added to the lagging strand. RNA polymerase 1 replaces the RNA primers with DNA primers. And then DNA ligase seals together the fragments, the Okazaki fragments, to make a long, continuous, complementary strand of DNA here. All right, so as you're looking at this, you're going to need to know the major enzymes that are involved, the roles that each of these enzymes play. You should be able to explain what an origin of replication is, explain the lagging strand and the leading strand, and the replication bubble. You should also know if the DNA, why DNA replication is semi-conservative. All right, proofreading and repair. So DNA polymerases also proofread as bases are added. They're checking to make sure that there's no mistakes or mutations that are occurring. Errors do happen, but they're quite rare. Pairing errors happen in 1 out of 100,000 nucleotides. We also have mismatch repair, special enzymes that fix incorrect pairings. So nucleotide excision repair, there's a nuclease. This is an enzyme, right? It ends in ASE. Nucleases cut out damaged bits of DNA. Oops, too far. And then DNA polymerase and ligase come in to fill in the gaps, which is pretty cool. So to excise means to cut out, right? Nucleotide excision repair is when they cut out damaged DNA parts and replace them with new copies that are correct. All right, so a problem at the 5' end. If DNA polymerase only adds nucleotides to the 3' end, there's no way to complete the 5' ends of the daughter strands. And what happens is over many, many replications, DNA strands grow shorter and shorter on the 5' end. So you see here where the lagging strand ends on the 5' end, there's an RNA primer. RNA primers are removed and replaced with DNA where a 3' end is available, but there's not at this 5' end. So it's shorter than the 3' end. After another round of replication, it gets shorter and shorter each time it replicates. Thankfully, these ending parts of DNA are a structure called telomeres. Now, telomeres are repeated units of short nucleotide sequences. So things that repeat at the end of the DNA. Here you can see the chromosomes. The telomeres are stained orange at the ends of these mouse chromosomes. So telomeres cap the ends of DNA to postpone the erosion of genes. So if enough replications occur, the telomeres at that 3' end will eventually erode or be eaten away, and it will start cutting into the functional part of the genes on the chromosomes. So telomeres are another example of non-coding DNA that serve an important function. They help protect the genes on the chromosomes. Telomerase is an enzyme that adds to telomeres. Eukaryotic germ cells have these, so the cells that divide over and over to produce sperm and egg. And also cancer cells have telomerase. Remember, cancer is just uncontrolled cell division. There's a lot of DNA replication that happens in cancer cells. It's interesting that cancer cells also have the ability to produce this enzyme. All right, so telomeres and telomerase. We have our eukaryotic DNA. There's a telomere at each end. We have our repeated units here. So telomerase extends the 3' end of the DNA strand. Here, you can see it adding on to the 3'. The other strand is extended in the usual way by primase, DNA polymerase, and ligase. The result is a longer telomere with a 3' end overhang. So the primer is removed here. So when telomerase is present in germ cells and cancer cells, it beefs up the 3' end. All right. Okay, so that wraps up our discussion of DNA replication or structure and function. Same thing. Do make sure that you watch the animation, and I'll put a link to that in our content for this week. If you have any questions about DNA structure and function or replication, make sure you write them down. bring them to student Zoom hours or office hours, or take them to class so we can discuss them together. All right.